In this video, we are going to discuss about the absolute configuration of amino acids. Now, amino acid has a chiral carbon in it. Okay, except glycine, all other amino acids have chiral carbon, and because of that, the amino acid exists in two configurations which are non-superimposable mirror images of each other. And these two configurations into which this amino acid exists because of that chiral carbon are known as enantiomers. Okay, these two configurations are known as enantiomers. Okay, these enantiomers are the two configurations of amino acids which are non-superimposable mirror images of each other. Now, this enantiomer can be identified by its absolute configuration. For example, glyceraldehyde has two configurations. When the hydroxyl group attached to the chiral carbon of this glyceraldehyde is at the left position with respect to the Fischer projection, then that is L configuration, and if the hydroxyl group is in the right position, then that is D configuration. Okay, if the hydroxyl group is at left, then that is known as L configuration, and if the hydroxyl group is at right, then that is known as D configuration. So in this figure you can see this is L-glyceraldehyde because the hydroxyl group is at the left position, and this is D-glyceraldehyde. The hydroxyl group is in the right position here. Now the prefix D and L in this diagram are representing the absolute configuration of glyceraldehyde. Now in this way, the absolute configuration of the amino acids is also specified by D and L system. Okay, with the help of this DLN system, we specified the absolute configuration of glyceraldehyde. In similar way, in the same way, the absolute configuration of amino acid is also specified by D and L system. Now, the designation of this D or L to an amino acid refers to its absolute configuration relative to the structure of D and L glyceraldehyde respectively. Okay, we take reference of D and L glyceraldehyde and then give or designate D and L absolute configuration to the amino acid. So this is L-alanine and this is D-alanine. Okay, so in the case of glyceraldehyde, we considered the OH group. Now we are considering the NH3 plus group, and if the NH3 plus group is at left, then that is L-alanine, and if this NH3 plus group is in the right position, then that is referred to as D-alanine, and that is how we determine or designate the absolute configuration of these amino acids. Now, all amino acids have two different enantiomeric forms, okay, except glycine. It cannot happen in the case of glycine, but except glycine, all amino acids have two different enantiomeric forms. Now, this is an interesting fact. Okay, all amino acids which are ribosomically incorporated into proteins have L configuration. And that is the reason why they are it is said that proteins contain L-alpha amino acids. Okay, all amino acids which are ribosomically incorporated, okay, with the help of ribosome, they are joined to form protein. Those amino acids have L configuration, and that is the reason why all your proteins contain L-alpha amino acids. Now, the basis for this preference of L-amino acid is not known, but it is very important to remember that all your proteins contains L-alpha amino acids. There is no D-alpha amino acid, it is only L-alpha amino acid in your proteins. Okay, so D form of amino acids are not found in ribosomically synthesized proteins. Now this D form of amino acids are found in some peptide antibiotics, and they are found in peptidoglycan cell wall of few bacteria. Okay, so D form of amino acid is not found in your ribosomically synthesized protein, but they are found in some peptide antibiotics and in the peptidoglycan cell wall of few bacteria. Now there is a second absolute configuration notation which uses the symbol R and S. R stands for rectus, which means right in Latin, and S stands for sinister, which means left in Latin. Okay, so this absolute configuration notation which uses symbol R and S can be used in case of proteins. Okay, that is your second absolute configuration notation. Now in this approach, the substituents that are attached to the asymmetric carbon, that is chiral carbon with 4 different constituents, these substituents are prioritized by the decreasing atomic number. Okay, the substituents attached to the asymmetric carbon are prioritized according to their decreasing atomic number. So the atoms with higher atomic number are ranked above the atoms which have lower atomic number. Okay, that is how this second absolute configuration notation works. Okay, the substituents are prioritized according to their decreasing atomic number. For example, oxygen of the OH group takes precedence over the carbon atom which is attached to the CH3 group. Okay, carbon atom of the CH3 group that are bonded to some chiral carbon atom. Okay, if you are considering OH group and CH3 group, then the oxygen of the OH group will take the precedence over the carbon atom of the CH3 group in this type of configuration. Now if any first substituent atoms are of same elements, then the priority of the groups is decided by the atomic number of the second or third or fourth or fifth atom that is outward towards the chiral carbon atom. Okay, if the first substituents atom are the same, then we look for the second atom and third atom's atomic number, and then we decide the priority of the group. Okay, so for example, this CH2OH group takes precedence over the CH3 group. Okay, the first atom is carbon. Here also first carbon is atom. The second two are hydrogen in both of them, but the third one here is oxygen, and in this case it is hydrogen, so you give priority to oxygen, and that is how this CH2OH group takes precedence over CH3 group. After you give the priorities to the group, the prioritized groups are assigned with letters such as W, X, Y, Z, and the order of priority goes like this. W has the highest, then X, then Y, and then Z. So in the 4 substituents that are attached, the group which has the highest priority, you designate it with letter W, and in same way the group which has the lowest priority is designated with Z letter. Now the configuration is assigned by looking down for the bond which has the lowest priority substituents, that is Z. Okay, we look down for a bond which has the lowest priority substituent, and on the basis of that we assign the configuration. So the order of the group, that is W, X, Y, if this order is in the clockwise direction, then the configuration of the chiral center is designated as R, and if the order of the group W, X, Y, this order of the group is in counterclockwise direction, then the chiral center is designated as S. Okay, the configuration of the chiral center is designated as if the order of the group is in the clockwise direction, counterclockwise direction, and the configuration of the chiral center is designated as R if the order of the group is in the clockwise direction. So this is D-glyceraldehyde and R-glyceraldehyde. As you can see, we have given the priority to the group. That is simple. So OH has the highest priority, then X, then CHO has, and then CH2OH has, and at last Z. Okay, so this is how the priority goes. So, and this is representing that the order of the group is from W, X to Y in the clockwise direction, and that is the reason why this is designated as R-glyceraldehyde. Okay, and in this case you can see it is the order of the groups is in the counterclockwise direction, okay, in this direction, and that is the reason why this compound is designated as S-glyceraldehyde. Okay, so similar case in case of an amino acid, that is alanine. As you can see, the highest priority is given to the NH3, then COO minus, then CH3, and at last H. So that is how the priority is given, and the order of group is WXY. This order of group is in the clockwise direction, and that is the reason why this alanine is referred to as R-alanine. Okay, so that is how that absolute configuration notation using the symbol R and S works. Now the absolute configuration of amino acid having alpha carbon is designated by D and L system and not by modern R and S system. Okay, the system we just described above is not used. The DLN system is used to explain the absolute configuration of the amino acids having alpha carbon. Now, according to this RS system, all L amino acids which form protein are S amino acids. Okay, they have S configuration. Okay, all amino acids have S configuration which make proteins, except L-cysteine. Okay, in this case, L-cysteine is exception. Okay, the L-cysteine according to RS system is R-cysteine. Okay, so all amino acids which make protein are in the S configuration except this L-cysteine which is in the R configuration. So thank you for watching this video. See you in the next video. That is what I can say.